Good morning, everyone, and welcome to today's presentation. I'm Dr. S.K. Singh, and I hold a PhD in this field. Today, I will be discussing the qualification of the Total Organic Carbon, or TOC, analyzer. Over the course of this presentation, we'll explore the key aspects of TOC analyzers, their importance, and how they are qualified for accurate measurements. I look forward to sharing insights with you all. Let's get started. In this slide, I want to walk you through the scope of our project. First, we're focusing on the qualification and validation of the TOC method, specifically for water systems and cleaning rinse or swab samples. Our goal is to ensure that the instruments we use perform reliably and meet the criteria set by pharmacopoeial standards. We are covering different types of samples, including purified water, water for injection, and rinses or swabs from cleaning processes. These samples are critical because they can directly impact product quality and safety. Now let's clarify what TOC is. Total Organic Carbon, or TOC, measures the total amount of organic carbon present in a sample after it has been oxidized to carbon dioxide. It's a key indicator of organic contamination, especially in water systems. In the context of pharmaceutical manufacturing, TOC is a critical quality metric. It's used to evaluate purified water, WFI, and cleaning validation processes, as well as environmental monitoring. Essentially, it helps us ensure that our water systems are clean and that our cleaning procedures are effective, safeguarding product quality and patient safety. Let's take a moment to look at some of the applicable guidelines that govern our work in this area. First, we have USP 643, which focuses on total organic carbon, or TOC, specifically in pharmaceutical water systems like purified water and WFI. This guideline helps us ensure the purity and cleanliness of the water used in manufacturing. Next, we have the European Pharmacopoeia, specifically EP 2244, which also sets standards for water quality and TOC measurement. These regulations provide a solid framework for maintaining consistent water quality across different regions. Then there's the ICH Q2R1, which outlines the principles of analytical validation, things like precision, accuracy, and linearity. These principles are essential because they ensure our methods are reliable and reproducible. When we talk about TOC, we're referring to total organic carbon, which is a critical parameter in assessing water purity. Monitoring TOC levels helps us detect any organic contaminants that could compromise product quality. Additionally, we should be aware of GMP references, such as FDA 21 CFR Part 211 and EU GMP Annex 15. These regulations reinforce the importance of validation and control in pharmaceutical manufacturing, including water systems. Overall, these guidelines form the foundation of our quality assurance practices, ensuring that we maintain high standards and consistency in our processes. Let's take a closer look at the basics of TOC analyzers. The core measurement principle involves organic carbon, or OC, which is first oxidized to produce carbon dioxide, CO2. Once we've got CO2, we detect it using common methods like non-dispersive infrared, or NDIR, or sometimes conductivity detection. The amount of detected CO2 then allows us to report the total organic carbon, or TOC, in the sample. It's also helpful to understand how TOC can be derived. Essentially, TOC equals the total carbon, TC, minus the inorganic carbon, TIC. So, by measuring TC and TIC separately, we can determine the organic portion. This process is fundamental because it provides a clear picture of organic contamination or organic content in water samples, which ties directly into our overall theme of ensuring water quality and process efficiency. Let's take a look at the qualification life cycle, which is essential for ensuring that our systems are functioning correctly and reliably. It consists of three key phases: IQ, OQ, and PQ. First, we have installation qualification, or IQ. This phase is all about verifying that the system has been installed according to the manufacturer's specifications. It's like a checklist to make sure everything is in the right place and set up properly. Next is operational qualification, or OQ. Here, we test the system's performance across the expected operating range. This step ensures that the equipment performs consistently and reliably under normal conditions. Finally, we have performance qualification, or PQ. This is where we confirm that the system meets all acceptance criteria during routine operation. It's about validating that everything continues to work as it should in real-world, everyday use. Connecting this to our overall theme, these phases help us maintain quality, compliance, and confidence in our processes. They form a structured approach to validating our systems from installation through routine operation, ensuring ongoing quality and performance. Let's take a closer look at this example calibration plot. What we're seeing here is a graph that plots the signal response against known parts per billion, or ppb, concentrations. The key point to remember is that we require an R-squared value of at least 0.99, as specified in the ICH Q2 guidelines, to ensure our calibration is reliable and accurate. Moving on to the calibration procedure itself, it involves using standards that are critical for accuracy. These standards include a TOC-free blank, which helps us establish a true baseline, and certified carbon standards, such as potassium hydrogen phthalate, or KHP, or sucrose. These standards are essential because they provide a known reference, allowing us to generate a calibration curve that truly reflects the relationship between signal and concentration. Overall, the goal is to develop a calibration that is both precise and reproducible, which is fundamental for ensuring the validity of our analytical results throughout the process. Now, let's look at how we calculate the corrected responses. First, we take our raw sucrose measurement, which is 0.51, and subtract the blank correction of 0.02, giving us a corrected sucrose value of 0.49. Similarly, for benzoquinone, we start with 0.495 and subtract the same blank correction of 0.02, resulting in 0.475. Next, to find the percentage response, we divide the corrected benzoquinone response by the corrected sucrose response, then multiply by 100. So, 0.475 divided by 0.49 times 100 equals approximately 96.9%. Since this is within the acceptable range of 85 to 115%, we can say that the test passes. Now moving on to the SSD calculation, or system suitability test, which is a key quality control step. Here, we measure three responses: the blank, the sucrose standard, and the benzoquinone. The blank, R left paren W right paren, is 0.02 mg per liter. The sucrose standard, R left paren S right paren, is 0.51 mg per liter. And the benzoquinone response, R left paren SS right paren, is 0.495 mg per liter. Using these, we calculate the response efficiency with the formula: 100 times the difference between R left paren SS right paren and R left paren W right paren, divided by the difference between R left paren S right paren and R left paren W right paren. This tells us how effectively our system responds to the analyte. Finally, the acceptance criteria for this test are between 85 and 115%, which corresponds to a plus or minus 15% range. Meeting this ensures our system is suitable for reliable analysis, maintaining confidence in our results and ensuring quality control throughout the process. Now, let's take a closer look at the example linearity data. Here, we're examining how well our measurement method performs across a range of concentrations. The goal is to ensure that our method provides consistent and proportional results as the analyte levels change. Validation parameters are key in this process. They help us confirm that our method is reliable and accurate. For example, we look at the linearity itself, but also other factors like accuracy, precision, and detection limits. By thoroughly evaluating these parameters, we can be confident in our method's performance, which is crucial for producing trustworthy results. This step is essential because it ties into the overall theme of method validation, making sure our analytical method is both robust and dependable in real-world applications. Now, let's take a closer look at how we derive TOC limits during cleaning validation. The key idea here is that the TOC limit from cleaning is often based on a simple but important formula. Essentially, we calculate the TOC limit in micrograms per square centimeter by multiplying the maximum allowable carryover, or MACO, by the dilution factor, and then dividing that product by the surface area of the equipment contact surface. The MACO, or maximum allowable carryover, represents the highest amount of residue that we consider safe to leave behind without impacting product quality or safety. The dilution factor accounts for how much the residue is diluted during subsequent processing steps, which is crucial for ensuring the residual levels are within acceptable limits. And finally, the surface area is simply the size of the equipment contact surface. The larger the surface, the more potential residue there could be, so this helps us normalize our limits accordingly. Connecting these points, this formula provides a systematic way to set realistic and scientifically justified TOC limits that help us maintain product safety and quality throughout the manufacturing process. It's a fundamental part of establishing a robust cleaning validation program. Now, let's take a closer look at the key parameters involved in cleaning validation. These are essential because they ensure that our cleaning processes are effective and consistent. First, we have to focus on the asterisk residue limits asterisk, which are the maximum allowable amounts of residual contaminants after cleaning. Setting these limits correctly is crucial to prevent cross-contamination and ensure patient safety. Next, we consider *** sampling methods ***, how and where we collect samples to verify cleanliness. It's important to choose representative sampling points and techniques to get accurate results. Then, there's *** analytical methods ***, which refer to the specific tests used to detect residues. These methods must be validated for accuracy, sensitivity, and reliability. Another key parameter is ** frequency of validation, meaning how often we perform these checks. Regular validation helps us maintain control over cleaning processes over time. Finally, we need to monitor ** cleaning parameters like temperature, pH, and contact time during the process itself, because these factors directly influence cleaning effectiveness. All these parameters are interconnected and play a vital role in creating a robust cleaning validation program. They help us ensure our facilities are compliant, safe, and capable of producing high-quality products consistently. Let's take a closer look at the concept of MACO, which stands for Maximum Allowable Carryover. This is a crucial calculation in our overall framework because it helps us determine the highest amount of certain substances or residues that can be safely carried over from one process or batch to the next. Understanding MACO is important because it directly impacts compliance with safety standards and ensures that we maintain product quality. When we calculate MACO, we're essentially setting a limit, one that balances safety, efficiency, and regulatory requirements. It's also worth noting that this calculation ties into our broader goal of maintaining control over our processes. By establishing these limits up front, we can better manage risks and make informed decisions about process adjustments or quality checks. In summary, MACO is a key metric that helps us ensure safety and quality across our operations, and mastering this calculation is fundamental to achieving consistent, compliant results. Now, let's take a closer look at the calculation of the TOC surface limit, which is measured in micrograms per square centimeter, or microg/cm2. This metric is critical because it helps us understand the maximum allowable amount of total organic carbon that can be present on a surface without posing a risk. When we talk about the surface limit, we're essentially setting a threshold to ensure safety and compliance, especially in environments where contamination control is vital. Calculating this limit involves considering various factors, including the type of surface, its use, and the potential for contamination transfer. What's important to remember here is that these calculations aren't just numbers. They represent a safeguard, helping us establish standards that protect both health and integrity of the materials or environments we're working with. By understanding and applying these surface limits accurately, we can better manage cleanliness and contamination, aligning with our overall goal of maintaining strict quality and safety standards across the board. Let's now focus on documentation and reports, which are essential for ensuring our data's integrity and reproducibility. First, we include calibration curves, which are fundamental for establishing the relationship between our instrument response and the analyte concentration. Next, we document SSD results, providing us with critical information about the sample stability and consistency. We also prepare a validation summary that covers linearity, precision, accuracy, and limits of detection and quantification. These parameters help us confirm that our method is reliable and fit for purpose. Additionally, we keep raw data and calculations, which are vital for transparency and troubleshooting if needed. Finally, traceability to standards and instrument logs is key. It links our measurements back to recognized standards and ensures proper instrument maintenance and calibration history. All of these components together support the robustness of our analytical process and ensure that our data stands up to scrutiny, aligning with the overall goal of producing trustworthy and high-quality results. Let's talk about routine monitoring and requalification. First, it's important to understand that routine checks are a key part of maintaining quality and compliance. We perform daily or shift-specific QC checks to ensure everything is functioning properly on a regular basis. In addition, quarterly calibration checks are necessary to keep our equipment accurate and reliable over time. Whenever we perform maintenance or experience any failures, that's the time to reevaluate and verify that our systems are still performing as expected. Now for requalification, which includes operational qualification or OQ and performance qualification or PQ. This is typically done annually. However, if there are any significant changes through change control processes, this schedule might need to be adjusted to an earlier requalification. All these steps are essential to ensure ongoing compliance, optimal performance, and ultimately, product quality. By following this schedule, we're proactively catching issues before they impact our results, which ties back to the overall theme of maintaining high standards in our operations. Let's wrap up with some key conclusions and compliance points. First, our qualified TOC system fully meets USB and EP criteria, which is essential for ensuring the quality and reliability of our testing processes. This means we're aligning with recognized industry standards. Next, we've validated critical parameters like linearity, accuracy, and precision. These validations confirm that our system produces consistent and accurate results across different conditions and sample concentrations. Additionally, we have documented both the sample suitability test, or SSD, and routine checks. This documentation is vital for maintaining traceability and demonstrating ongoing compliance over time. All of these elements work together to ensure that we can reliably detect organics, which is crucial for product safety and quality. Plus, they help us stay compliant with GMP regulations, ensuring our processes meet all necessary standards. In summary, these points reinforce the robustness of our system and our commitment to quality and compliance throughout the process. For further support, please feel free to contact us. We're here to help you with any questions or additional assistance you might need. Don't hesitate to reach out. Your success is our priority. Thank you so much for your time and attention today.